Hello everyone, good afternoon, how are you all? I hope each and every one of you is in the pink of health and happiness. Shall we start the class? Today we are going to learn about biotechnology principle and process. Understanding, as usual, we are going through the MCQs. Along with that, we are going through the concepts as well. Okay, so our first question is, in a chromosome, there is a specific DNA sequence responsible for initiating replication. It is called easy. It is yes, origin of replication. That is ORI site. D for Delhi is the correct answer. What is the fate of piece of DNA? We are telling about piece of DNA. Okay, piece of DNA carrying a gene of interest, which is transferred to an alien organism. So, what is the fate of piece of DNA? We need to find the correct answer. The piece of DNA would be able to multiply itself independently in a progeny cells of the organism. Just a piece of DNA would be able to multiply itself independently? No. It may get integrated in the genome of recipient? Yes, it may. And it may multiply and be inherited along with the host DNA? It may multiply and be inherited along with the host DNA? Yes. The alien piece of DNA is not an integral part of the chromosome. If it is not the integral part of the chromosome, it will not multiply. It shows ability to replicate. If it is in the integrated part, it will show, I mean, like just a piece of DNA, it will not show the ability to replicate. If we see, B and C are the correct options, which means A is the correct option. B and C are the correct statements. Pathogenic bacteria gain resistance to antibiotics due to changes in their yes, definitely in their plasmids. Plasmid have this antibiotic resistant site, right? Okay, which of the following methods is not commonly used for introducing foreign DNA into plant cell, which is not common. Okay, not common. Underline wherever not or incorrect is there. Just tell me not common. Bacteriophages. Yes. So, which means agrobacter mediated. This is very common. Gene gun is common in among plants, and disarmed pathogen as a vectors is also, but as bacteriophages are not common. The cutting of DNA at specific location became possible with the discovery of
restriction enzyme.
Yes, restriction. D for Delhi is a correct answer as a restriction enzymes. Yes, tell me. Now coming to the tools of recombinant DNA technology, identify the incorrect statement related to gel electrophoresis. Incorrect, we need to find out. Separated DNA fragments can be directly seen under the UV radiation. Can they directly be? We need to stain them, right? So that is a incorrect option. We need to find the incorrect. We got it. First A is an incorrect option. We will go through others whether they are correct or not. Separated DNA can be extracted from the gel piece. Yes. Fragment of DNA moves toward the anode. Yes, because this DNA is negative, so it moves toward the anode. Sieving effect of agarose gel helps in separation of DNA fragments. That is also true. Okay, B, C, D are true, and as we already got the answer, A is a incorrect statement. That is the right answer. Recombinant DNA molecule can be created normally by cutting the vector DNA and source DNA respectively with, the vector DNA and the source DNA has to be cut respectively with same or similar type, same restriction enzyme, isn't it? So now here you need to find the same restriction enzyme. One is the DNA molecule of the vector and the source DNA. Which one? Which option is right? Same.
Well,
yes, obviously, no? Because it will create the ends which has to be again joined by the similar, like for example, if this is a vector DNA, like that, if it cuts, if it's cut, staggered ends, if it cuts, sorry, staggered end, the molecule, the source DNA should also be cut by the same restriction enzyme, right, for it to match these staggered ends, isn't it? So it has to be cut by the same. If the vector is cut by the hint 2, the source DNA should also be cut by the hint 2 only. Select the restriction endonuclease enzymes whose restriction site are present for tetracycline resistant gene in PBR 322 cloning vector. Tell me which is in tetracycline resistant. So PST is in ampicillin. So it is BAM H1 and SAL1. If you see, these restriction site are in the tetracycline resistant site, isn't it? Option A is a correct answer. Which of the following are correct about echo R1? Cut the DNA with blunt ends. Echo R1, cut the DNA with the staggered end. Cut the DNA with sticky ends or staggered end, correct. Recognize a specific palindromic sequence, that is, yes, that is GAA-TTC, and it cuts here in between G and A. Cut the DNA between the base G and A when encounters the DNA sequence GAA-TTC, correct. It is the exonuclease. No, it is the endonuclease, restriction endonuclease. So B, C, D are the correct is the correct answer. B, C, D statements are correct. D for Delhi is the correct answer. Understanding? Hint 2 always cuts DNA molecule at a particular point called restriction sequence, and it consists of how many base pairs? Hint 2.
6.
Yes, it consists of 6 base pairs. D for Delhi is the correct answer. Now the following diagram shows the restriction site in E. coli cloning vector PBR 322. Find the role of X and Y. Find the role of X and Y. What is this? It is ORI. And what is this? This is ROP. Tell me, the gene X is for protein involved in replication of plasmid. Gene X is not replication for plasmid, it is for the origin of replication. Origin, it is ORI. Gene X is responsible recognition sites and Y responsible antibiotic resistant? No. Gene X is responsible resistant to antibiotics? No. The gene X is responsible for controlling the copy number of the linked DNA, that is copy number is by the ORI site, definitely, okay, X is true. And Y for protein involved in replication of plasmid, ROP, that is also true. So it is D for Delhi is the correct answer. The TI plasmid of Agrobacterium tumefaciens stand for, TI means, tell me, tumor inducing plasmid. So with A as a, first option itself we got the correct answer, that is tumor inducing plasmid. Upon exposure to UV radiation, DNA stained with ethidium bromide will show which color? Bright orange color. A is the correct answer. In gene gun method used to introduce alien DNA into host cell, microparticles of dash metals are used. Silver, copper, zinc, tungsten or D for Delhi, that is tungsten and gold are the metals used. Which of the following is not a cloning vector? Bacterial artificial bacterial artificial chromosome, yeast artificial chromosome, PBR 22, all are the cloning vectors, but probe is not a cloning vector, isn't it? Probe is not a cloning vector. Ligation of foreign DNA at which of the following site will result in loss of tetracycline resistant of PBR322? BAMH1. This is a restriction site which is present in the tetracycline resistant site, isn't it? EcoR1 is outside, PVU and PST is in the ampicillin resistant site. Now match list 1 and 2. Kanamycin is antibiotic resistant. CLA1 is a restriction site. Disarmed retrovirus,
delivers
as a vectors, these are vectors. Kanamycin resistant gene is a selectable marker, isn't it? So we need to find D2. Okay, D2 is in this both, so you can remove these two. C1. C is the correct answer. Which of the following can act as molecular seizures? Which one we call it as molecular seizures? Restriction enzymes, we call it as molecular seizures. Easy. Given below are two statements, one is assertion reason, okay. When a particular restriction enzyme cuts strand of DNA, overhanging stretches or sticky ends are formed, isn't it?
Yes.
Yes, because some restriction enzyme cut the strand of DNA a little away from the center of the palindromic site. That's why we get the staggered end, or otherwise we call it as sticky ends. So this is also true. Is it the correct reason? Yes, the reason explains the assertion, right? So it is the correct explanation. So we can say both A and R are true or correct, and R is the correct explanation of A. Separation of DNA fragments is done by technique known as separation.
Gel.
Yes. Gel electrophoresis. In the following palindromic base sequence of DNA, which can be cut easily by particular restriction enzyme? The particular restriction enzyme they are talking about EcoR1. Tell me.
C.
GAATTC, CTTAAG. This is the correct answer. Which of the following is not a desirable feature of cloning vector? Not a desirable feature. Presence of two or more. Yes. What happens when there is a presence of two or more recognition site? Tell me.
Creates confusion.
Actually, it cuts the DNA into pieces, isn't it? For example, even in cloning vector, even in the vector, what happens if there is more than one? Okay, if this is a cloning vector, listen, if this is a cloning vector and if EcoR1 is present here, for example, EcoR1 is present here also, tell me, and it is present here also. So when we keep this EcoR1, this is a restriction site, if we do EcoR1 restriction enzyme, what happens to the vector here? The vector will be cut into pieces, right, like this. They cut into pieces, so many pieces. Will there be this still be vector, this one? It will be like cut into pieces. So it is not a good feature. Presence of two or more recognition site. It is not a good feature, isn't it? So this is wrong. I mean to say not a desirable character. Yes, this is not a desirable character. Presence of origin of replication, it needs to have the origin of replication. Presence of marker gene should be there. Presence of single restriction enzyme site, yes, single restriction enzyme site should be there. Given below are two statements. Statement 1, restriction endonucleases recognize specific sequence to cut DNA known as palindromic nucleotide sequence. True. Restriction endonucleases cut the DNA strand a little away from the center of the palindromic site.
Yes.
So both statement 1 and 2 are correct. B for Bombay is the correct answer. DNA strand on a gel stained with ethidium bromide when viewed under UV radiation appear as repeated question. Which color?
Orange.
Bright orange bands, yes. Plasmid PBR 322 has PST1 restriction enzyme site within gene ampicillin resistance that confers ampicillin resistance. Okay, this one. If this enzyme is used for inserting a gene for beta galactoside production and the recombinant plasmid is inserted in an E. coli strain, so what will be the consequence of it? The transformed cell will have the ability to resist ampicillin? No. If this particular resignation site is inside the ampicillin, it will lose the ampicillin resistance. It will lead to lysis of host cell? No. It will be able to produce the novel protein with dual ability? Doesn't explain anything. It will not be able to confer ampicillin resistance to the host cell. Yes. Understood? For example, this is PBR 322 and this is ampicillin resistance. So here PST1 is there here. Okay, if the enzyme is used to insert a gene of beta gal, if we insert a gene beta gal here, for example, with restriction endonuclease, we cut the DNA vector and we inserted a gene of interest, our gene of interest here, a gene of interest here. So will the ampicillin resistance still present? Will this bacteria give the ampicillin resistance if it is incubated in the ampicillin given medium? No, right? As this site lost its ability, it will not be able to confer ampicillin resistance to the host cell, isn't it? So D is the correct answer. A specific recognition sequence identified by endonucleases to make cuts at specific position within the DNA is palindromic sequence or palindromic nucleotide sequence. First discovered restriction endonuclease that always cuts the DNA molecule at particular point recognizes a specific sequence. That is Hind 2. Match 1 and 2. Okay. Bacillus thuringiensis. Yes, cry genes or cry proteins. Thermus aquaticus. DNA polymerase, Staph polymerase, we got it from that. Yes. Agrobacterium tumefaciens, cloning vector. Salmonella typhimurium, construction of first rDNA molecule. Yes. So 4, 2. Okay. We can remove these. Okay. And 3, 1. Okay. 2, 3. Or else if 1, 4. Okay. If it would have come from that side, we would have got the answer in the first match itself. 1 to 4. A is the correct answer. The sequence that controls the copy number of linked DNA in the vector is termed ORI. A is the correct answer. And the specific palindromic sequence which is recognized by ECO R1. GAATTC. Yes, that one.
Option D.
Option D. GAATTC, GAATTC. So this is the one. Identify the wrong statement with regard to restriction enzymes. They cut the strand of DNA at the palindrome sequence. They are useful in genetic engineering. The sticky ends can be joined using the DNA ligase. Each restriction enzyme functions by inspecting the length of DNA sequence. You need to find the wrong statement. They cut the strand of DNA at palindromic sequence. Identify the wrong statement with regard to restriction enzymes.
Option C.
Sticky, yes, sticky, okay. Sticky ends are ligated by, here anyway the hydrogen bonds will be formed by themselves actually here, and the sticky ends joined by the DNA ligase. What DNA ligase actually does, it actually bonds the bond between this phosphodiester bond, phosphodiester bond between sugar and phosphate. Sugar and phosphate. Sequence can be joined using the DNA ligase. No, actually what DNA ligase does, DNA ligase joins these, actually they joins the 2 DNA here, 2 DNA molecule in this particular site, in this particular site, in this particular site, where it establish the phosphodiester bond. Understanding? It established the phosphodiester bond, DNA ligase. It joins the DNA molecules, not the sticky ends. Sticky ends, these ends will be joined by the hydrogen bonds. This is also true. We need to find the wrong statement, that is C. Choose the correct pair from the following. Polymerases, no, this is wrong. Nuclease, separate the 2 strands of DNA. No. Exonucleases make cuts a specific position within the DNA, not within the DNA, it's from the end. That's also true. Ligases join the 2 DNA. Okay, this makes sense. D for Delhi makes sense. I'm really sorry for that. Yes, thank you, Sakshi. Ligase joins 2 DNA molecules, isn't it? Okay, polymerase anyway. Yes. Okay. So this is a correct pair. Choose a correct pair. Following statement describe the characteristics of the enzyme restriction endonuclease. Identify the incorrect statement. We need to find the incorrect statement. The enzyme cut DNA at identified position within the DNA. Which one is the incorrect one? Binds a specific. Okay. The enzyme binds the DNA at specific sites and cut only one of the 2 strands. This is wrong. It cuts both the strands. Yes. B is the incorrect statement. Option B is the incorrect statement. A gene whose expression helps to identify transformed cell is known as
Selectable marker.
selectable marker. With the help of selectable marker, we can identify which is transformants and non-transformants, or recombinants or non-recombinants. Restriction endonucleases are in fact were synthesized by bacteria as a part of their defense mechanism at first. That ability we are using it. So this is correct answer. Yes, very good. Which of the following restriction enzymes produce blunt ends? Blunt end among these are produced by EcoRV. Okay, it produces blunt ends. Blunt ends, in a sense, there are two types of ends. That is, one is staggered end, which means this one, like this, GAATTC. GAATTC. Here, in this, the restriction enzyme cuts the DNA here, right? Which means it will be, these are the overhanging, this particular part is the overhanging one, this particular part is the overhanging one. This is called sticky ends or staggered ends. Otherwise, if it is like this, GGCC, CCGG, if this is a sequence, 5 prime to 3 prime, 5 prime to 3 prime, if it cuts like this here in this particular manner, no overhanging ends, that is called blunt ends. The blunt ends, this is called blunt ends, and this is called staggered end. So EcoRV produces blunt ends. A foreign DNA and plasmid cut by the same restriction endonuclease can be joined to form a recombinant plasmid using joining.
Ligase.
Before Bombay, ligase. Which of the following is a restriction endonuclease? Protease, DNase, RNase are protease degrade protein, DNase, DNA, RNase, RNA. So these are not the restriction enzymes. Hint2 is a restriction enzyme which is first discovered, by the way. Which of the following is not a feature of plasmid? Not a feature of plasmid. Independent replication, yes. Circular structure, yes. Transferable, yes. Single stranded, the plasmid is double stranded. The plasmid is double stranded, not single stranded. So D for Delhi is not a feature of plasmid. The DNA molecule to which the gene of interest is integrated for cloning is called? For cloning, which one you will be used? Is a vector. Gene of interest is cloned. It is cloned to a vector. The introduction of tDNA into plants involve altering the pH of the soil, exposing the plant to cold and for a brief, allowing the plant roots to stand in water, infection with the, infection of the plant by Agrobacterium tumefaciens. D4 Delhi is the correct answer for introducing tDNA. Which vector can clone only a small fragment of DNA? For small fragment of DNA, we need to use plasmids. D4 Delhi is the correct answer. DNA fragments generated by the restriction endonuclease in a chemical reaction can be separated by electrophoresis. DNA can be separated, the individual strands, okay. It can be separated by gel electrophoresis. Now, process of recombinant DNA technology processes. Following other steps involved in the process of PCR. Okay, we need to find the correct sequence. First will be definitely denaturation, then annealing. Then for amplification, before that we treat with the Taq polymerase and deoxynucleotides. Yes, CA. Okay, we got it. Okay, with the first this thing we got the answer anyway. Yes, before that it will be treated with Taq polymerase 3, extension and amplification. Yes. A is the correct answer. Which of the following statement is incorrect? Bioreactors are used to produce small scale bacterial culture. Now actually they are used for large scale. So which means this is wrong. We need to find the incorrect one. We got it in the first statement itself. That is A is the incorrect option. I mean incorrect statement, A is the correct option. Which of the following statement is incorrect? Okay, the same one. During the purification process for recombinant DNA technology, addition of chilled ethanol precipitates out the
DNA.
It precipitates out DNA. Main steps in the formation of recombinant DNA are given below. Arrange these steps in correct sequence. Arrange it. It is cutting off DNA at the specific location by the restriction enzyme. Yes?
Cutting of DNA.
Yes. Then isolation of desired one. Insertion of recombinant DNA into the amplification of gene of interest using PCR. And insertion of recombinant DNA. BCDA. BCDA. B for Bombay is the correct answer. Thermostable DNA polymerase used in PCR was isolated from? It was isolated from Thermus aquaticus bacteria. The enzyme A is needed for isolating genetic material from plant cells, and enzyme B for isolating genetic material from fungus. Choose the correct pair. For plant it should be cellulase, and for fungus it will be chitinase. C is the correct answer. For plant cell it is, for isolating we need to break open the cell wall. In order to break open the cell wall, the plant cell is made up of cellulose, we need to use cellulase. The fungus cell wall is made up of chitin, we need to use chitinase to break open the cell wall, because inside that we can isolate the DNA. Refer the following statement for agarose electrophoresis. We need to find the most appropriate, which means correct option. Agarose is a natural polymer obtained from seaweed.
Yes.
Yes. The separation of DNA molecules in agarose electrophoresis depends on the size of DNA.
Yes.
The DNA migrates from negatively charged electrode to the positively charged electrode.
Yes.
DNA migrates from positively charged electrode to negatively charged electrode. No. This is true. A, B, C are correct. C is the correct option. Which one of the following statement is not true regarding gel electrophoresis technique? We need to find not true. Bright orange color bands of DNA can be observed in the gel when exposed to UV light. The process of extraction of separated DNA strands from gel is called elution. The separated DNA fragments are stained using etherium bromide. The presence of chromogenic substrates give blue colored DNA bands on the gel.
No.
So, D for Delhi is a wrong statement, not true regarding the gel electrophoresis. Which of the following, assertion reason, polymerase chain reaction is used in DNA amplification?
Yes.
Yes. The ampicillin resistant gene is used as selectable marker to check the transformation? Yes. But polymerase chain reaction and this is a selectable marker. Is it having any relation? Is it a correct explanation of A?
No.
No, it's not a correct explanation of A, which means both are true, but it is not a correct explanation of A. During the purification process of recombinant DNA technology, addition of chilled ethanol precipitates out. Repeated question. You already know the answer. A, DNA. It precipitates out DNA. During the process of gene amplification using PCR, if very high temperature is not maintained in the beginning, then which of the following step of PCR will be affected first? If it
Denaturation.
denaturation, for denaturation we need very high temperature. If very high temperature is not maintained, then it will affect the denaturation of DNA. In gel electrophoresis, separated DNA fragments can be visualized with the help of
Ethylene bromide in UV radiation.
ethidium bromide in UV radiation. Yes, this is a correct answer. Here ethidium bromide is there, not infrared. In a mixture, DNA fragments are separated by separated of DNA fragments is taken place by electrophoresis. Easy. A very easy chapter this one is. In recombinant DNA technology, antibiotics are used
Selectable mark.
as selectable markers in order to find which are transformants and non-transformants. Now match 1 and 2. Bioreactor, production of large quantities of product. Electrophoresis, separation of DNA fragment. PCR, amplification of nucleic acids. ELISA, detection of pathogen based on antigen antibody reactions. 4-3. Yes. So A is the correct answer. Spooling. Spooling is, what do you mean by spooling? Collection of? Yes. Not cutting. It is collection of isolated DNA. Collection. When we chill out, when we add chilled ethanol, the DNA precipitate out. The collection of that particular DNA which is isolated, we call it as spooling. Okay. Select the correct statement from the following. We need to find the correct statement. The polymerase enzyme joins the gene of interest and the vector DNA. Restriction enzyme digestion are performed by incubating purified DNA molecules with a restriction enzyme of optimum condition. PCR used for isolation and separation of gene of interest. Gel electrophoresis used for amplification of DNA. Tell me. Ligase joins the gene of interest, not polymerase. Gene of interest and the vector DNA is joined by ligase. The restriction enzyme the restriction in digestion are performed by incubating purified DNA molecule with restriction enzyme of optimum condition. This is true. PCR is used for isolation and separation of gene of interest. PCR is used for amplification. Gel electrophoresis is used for amplification. No. Gel electrophoresis is used for isolation and separation of gene of interest. So this is ulta. So these two are wrong. The only thing, we need to find the correct statement. The correct statement is B for Bombay is the correct statement. Which one of the following equipment is essentially required for growing microbes on larger scale for industrial production of enzymes? Bioreactor. DNA replication out of a mixture of biomolecules can be achieved by treatment with?
Chilled ethanol.
How many times has been repeated? This is the third time we are doing this question. The correct order of steps of polymerase chain reaction? Easy.
Denaturation, annealing, extinction.
Denaturation, annealing and extension. D for Delhi is the correct answer. What is the criterion for DNA fragment movement on aqueous gel electrophoresis? Larger fragment move farther? No. Smaller fragment size, the farther it moves? Yes, the farther it moves. Isn't it? Before Bombay is the correct answer. The process of separation and purification of expressed protein before marketing is called downstream processing. Before Bombay is the correct answer. The DNA fragment separated on an agarose gel can be visualized after staining with. This is also third or fourth time repeated. It's very much repeated question. It's ethidium bromide. Which of the following is not a component of downstream processing? Preservation, yes. Separation is. Purification, yes. Expression is not. So, which is not? They have asked. B for Bombay is the correct answer. Stir tank bioreactors have been designed for designed for availability of oxygen throughout the process, not for anaerobic. Purification, no. Addition of preservative, no. It is for the availability, making the oxygen available throughout the process. That's why stir tank bioreactors are designed. The Taq polymerase enzyme is obtained from. This is also a repeated question, second time repeating. That is A. Thermus aquaticus. Which of the following is not correctly matched for the organism and its cell wall degrading enzyme? Fungi, chitinase, bacteria lysosome, plant cell cellulase, algae, methylase. Isn't it? So, this is wrongly matched. D for Delhi is a wrongly matched one. Okay, this is the last question in this chapter. Okay, we did around 71 questions continuously, and thank you all for the good response. It was a good response. That's why we could able to complete 70 questions in about 50 minutes. In about 40 minutes, yes, in about 40 minutes. It was a good response.